Hello po, magandang tanghali po sa ating lahat. Yun. Kamusta po ang ating araw? Yan, busy-busy naman sila. Sir Brian, ako po'y hindi makapag-share kung magagawan po natin ang paraan. Ayun, pwede na po.
Nakapag-post na ng link, YouTube link sa sa Facebook group.
Nakapag-post na.
Hinihingal ka lang, may oras pang natitira, kahit parang ang layo pa, habol. Kung harangan ka man, sumalakay man mga bantay, lahat kami maghihintay. Habol, habol, dehado kung dehado, para saan pang magagalos mo kung titiklop ka lang, titiklop ka lang. Matalo kung matalo, huwag ka sanang magkakamali, subuko na lang. Maagawan ka man, lalong huwag kang papipiga, kumpiyansa lang bawat bangga. Kung maripas ka na, kumanda ka na sa paglipad, pakpak natin ilalantad. Dehado kung dehado, para saan pang managalos mo kung titiklop ka lang, titiklop ka lang. Matalo kung matalo, huwag ka sanang magkakamali, subuko na lang. Ang puso ialay sa laban, kapalit ay tagumpay. Ang puso
Lawrence, may PM ako sa'yo.
Ay, nakikita mo? Ate. Ate kami korta ate.
Yan, magandang tanghali po sa lahat. Magandang tanghali.
Good afternoon everyone. First lecture po natin, nandito na po sa Calagua. So as of now, meron na po tayong 20 participants. Wait na lang po natin yung iba.
And good afternoon, everyone. We acknowledge the presence of our speaker for today, Miss Jonna Maika Lagwa. Yan. Welcome po ate. Welcome po ma'am sa ating pre-review session.
Liwanag sa
So good afternoon po uli. Pag naka 30 participants na po tayo, siguro start na po tayo.
Isa ko wala ko ni. I-unmute ba siya ba? Saan mo dito kabalo-bal ko ba ba?
At the right place at the right time, two roads intertwine. And if the universe conspired to meld our lives to make us fuel and fire, then know wherever you will be, so too shall I be. Close your eyes, dry your tears, 'cause when nothing seems clear, you'll be safe here from the sheer weight of your doubts and fears. Weary heart, you'll be safe here. Remember how we laughed until we cried at the most stupid things, like we were so high, but love was all that we were on. We belong. And though the world would never understand, when nobody hears you scream, I'll scream with you. You'll be safe here. Oh, save your eyes from your tears when everything
Again, good afternoon everyone. Welcome to Padayon Hiraya Manawari Free Layer Review Sessions. Okay, ngayon po meron na tayong 36 participants. Pwede nyo po bang i-type sa chat box kung taga saan po tayo mga university? So, mostly po nung mga participants natin ay from CVSU, UPLB, USM, and EU. Ayun po, si Ma'am Marinel from the Ilocos Sur Polytechnic State College, Santa Maria Campus. Si Sir Brian Manalastas from Cavite State. Si Sir Matthew Ray Licodin at DLSAU. Sir Renz, University of Southern Mindanao. USM, CMU, Cebu Technological University, NEUST, University of Negros Occidental, Recoletos, Visayas State University, Isabela State University. Good afternoon, everyone. So, proceed na po tayo sa introduction, no? We are a group of students, instructors, board exam takers, LEA top-notchers, and other professionals in the field of agriculture who are willing to share their knowledge of agriculture. Sir Renz? Oh, before I proceed pala, let me introduce myself first, no? I'm Rian Panerio, your moderator for today, and together with me is Mr. Renz J. Caducoy. Hi, Renz. Are you there?
Hello po. Good afternoon po sa ating lahat.
Ayun. So, ano nga ba ang ibig sabihin ng padayon, no? Ang ibig sabihin po nito is to continue or to keep going. And yung Hiraya Manawari naman po ay may the wishes of your heart be granted. And alam ko naman po lahat na nandito may wish para sa gaganapin na exam for November. So, next slide please. Ayun. So, idagdag ko na lang din po na this session is in partnership with CVSU K-FENAR Student Council, Matayuyan Crop Science Society, UP Agricultural Society, UP Animal Science Society, UP College of Agriculture and Food Science Student Council, UP League of Agricultural Biotechnology Students, KATSU or Catanduanes State University, College of Agriculture and Fisheries Student Body Organization, Anahaw Laguna, and 4-H Club Cavite. Thank you to our dearest partners for choosing us.
So yan, kung ready na po tayong lahat, ay atin pong buksan ang ating mga camera para sa ating picture taking. Huwag po kayong mahiya dahil lahat naman po tayo ay ginawang maganda at gwapo ng Diyos. Yun. So, open na po para meron po kaming documentation. Yun. So, wait lamang po. Yan. So, kung ready na ang lahat, sige, sabay-sabay tayo. Okay, ready? 1, 2, 3, smile! Yan. Isa pa po. Baka gusto niyo po na, wait lang, isa pa po. Yan. Okay, 1, 2, 3, smile. And last, wacky. Yan, ganito pala sa akin. Tatlo. You ready? 1, 2, 3, smile. Yun. Maraming salamat po.
So just to give recap lang ng previous session natin, yung una pong topic natin last week is yung plant growth regulation by Sir Elmer Real Montebon. Lecture 2 is tissue culture discussed by Sir Jose Diego Rojas. Yung third lecture ay sexual propagation by Sir Fredelino Vecina Jr. And Yung last lecture ay yung Asexual Propagation by Sir Felix Valdez. So, remind lang po namin ang lahat. So, naka-flash po sa screen nyo yung link ng evaluation form. Don't forget po na magsagot ng evaluation form para malaman din po ng mga organizers kung saan pa ba dapat mag-improve. And also, para makakuha or mabigyan ng copy ng review materials for today's session. So, dahil free itong review session at maiwasan ng pag-leak ng review materials, invite na lang po natin yung mga kakilala natin na mag-join or mag-message po sa page. Basta lagi po tayong mag-invite pag may post po na may panibagong set of lectures, share lang po natin para mas dumami pa po yung community natin. No? Dahil libre naman po ito. Hindi po kami madamot. So, ayun.
Yes naman.
Okay.
Ay, at saka isa pa pala, a reminder po na ang ating pre-test, o di ba? Saan kayo nakahanap ng free review na may
Yes.
pre-test? Di ba? Yun po ay pre-prepare ng mga top-notchers. So, ibig sabihin po ay sinala po namin ng maayos kung ano sa tingin namin ang lalabas o lumabas nung board exam. So, yun po ay aming pinagsama-sama at yun po ay aming pre-test. So, nawa ay itry po nating sagutan dahil napaka-sulit ng inyong free review session kung atin po yung gagamitin, di ba? Sa kinukanap nun, may free test, may free pre-test, tapos meron namang post-test, o di ba? Saan na kayo? Dito lang yan.
Dito lang yan sa Padayon
Center. Yes, Padayon. And of course, huwag kayong mag-alala dahil ang top 1 ay may papremyo. So saan pa kayo? Dito lang yan. May pa-pretest sila sa iba pero walang pa-premyo. Kami dito, kami pa yung libre. Kami pa nagpapapremyo. Oh.
Siyempre, sagot
diba?
yan ni Lance.
For now, ako muna ang sasagot. Oo. At saka naman, one time lang po kayo dapat sumagot ng pretest. Dahil po ang pretest at pang-isa, wag niyo naman pong damihan dahil hindi na po natin alam kung may natutunan ba tayo or na-memorize lang po natin. 'Di ba? So, I admit naman na nag-open notes ako pero bad 'yon. So, yun lang po.
So, ayun, acknowledge lang uli natin yung 46 participants natin dito sa Zoom. Yung sa Facebook Live, may Facebook Live ba or sa YouTube?
YouTube na po tayo.
Ay,
social na po
hindi
tayo.
ko na kinaya. Improving na. So, ayan no, mag-house rules muna tayo. Nakakapanibago lang kasi 45 lang yung participants natin ngayon sa Zoom. O sa YouTube daw meron tayong 10 participants or viewers doon. So, house rules lang, no? Be on time. Siyempre, kahit kung sa exam, hindi naman pwedeng pa-late-late tayo. Tapos kung hindi naman po magsasalita, paki-mute na lang po yung mic natin and Q&A after each talk po. And makinig po tayo maigi or mag-take down notes tayo. So, have fun learning. So, idagdag ko na rin po, ipalike na rin po natin sa mga kakilala natin yung Facebook group, Facebook page ng Padayan Hiraya Manawari. Ayun. So, proceed na po tayo. So, to introduce our first lecturer for this session, the Genotype and Environment Interaction. Our lecturer for today is a graduate of Bachelor of Science in Biology, major in Genetics in UPLB, and also a graduate of Master of Science in Genetics, major in Genetics, minor in Molecular Biology and Biotechnology, also in UP Los Baños. So let us welcome Miss Jonna Mika Lagwa. The screen is now yours, ma'am. Thank you so much.
Hello everyone, good afternoon. Can you hear me well? Can you hear me well?
Medyo po mahina po, ma'am.
Better? Okay na?
Opo.
Okay. So, thank you for inviting me here. And I think some of you, the participants from Cavite State University, has been my student na rin. So this will serve as a review as well of your basic genetics. So let me just share my screen first. A moment, please. Alright. Kita po ba? Can you see my screen?
Yes po.
Alright.
Yes po.
That's good. So for today, my topic is about genotype and environment interaction. Pero hindi natin maaalis that we will be having a brief discussion with regards to the basic genetics, the one that you have encountered in your, in I think second year, right, in your college. So before we start, let's have the definition of terms, okay? So some of this you have already encountered in the past lectures, and some of this you have encountered as well in your basic genetics. So para mas madali yung ating discussion later, let's define this first. So let's start with the gene, okay? So the gene is the basic physical unit of inheritance. So basically, okay, this is the information that is encoded in your DNA. Okay? And the DNA, the deoxyribonucleic acid, as it condenses, diba, you will have the chromosome. Okay? And there are specific parts of the chromosome wherein a specific gene is located. So that's what you call the, anong tawag doon? The specific address of a gene in a chromosome. So let me just see the chat. Okay, good. Locus. Okay, so yeah, that's the locus. Okay, that's the address in which a specific gene is located in a chromosome. Right? Next, we'll go to the allele. So what is an allele? An allele is a form of a gene. Okay, so a particular gene has certain forms, okay, different forms, be it two forms of a particular gene, three, four, okay? So that's what you call an allele, okay? So next naman is the genotype, okay? So when you say a genotype, this is an individual's collection of genes, okay? And at the same time, we also refer to a gene pair, okay, that is composed of two alleles as a genotype as well. Okay? So for us humans, we are diploid organisms, right? Our chromosome number is, we have two sets of chromosomes, one from the mother and one from your father. So in our chromosomes, okay, so we have two chromosome 1, chromosome 2, chromosome 3, and each of these chromosomes contain with them a specific allele for a particular gene. All right? So for us, let's say for example, the gene is, okay? So when we say, for example, you have the gene is symbolized by A, letter A, okay? So dito yung mga encounter yung homozygous, diba? Heterozygous, okay? When we say homozygous, class, I'm just refreshing your memory with this. So yeah, when you say homozygous, you have identical alleles, okay, in a gene pair. So let's say these are, sorry. So when we say you have homozygous, okay, so you have the same allele in a gene pair. Pag naman heterozygote or heterozygous, So you have different alleles in a gene pair. So that is the genotype. Okay? So for the phenotype naman, this is an individual's observable trait. Okay? So ito na yung nakita natin. Okay, the color of her hair, her skin, right? And even those that could not be observed readily, like our blood type, okay, A, B, and O. These are considered as phenotype as well. And next is the environment. So ano naman yung environment? The environment are all the things apart from your genes, from what is encoded in your DNA. Okay, so we consider that as the environment. Okay? And we can we can also differentiate them as macro environment or micro environment. So for macro environment, those that are common to a given location at a given time. Okay? So let's say temperature. Okay? So all individuals of a population are experiencing that particular environment, okay, or environmental factor, be it amount of rainfall. Okay, so those are parts of microenvironment. Pag naman microenvironment, ito naman is the environment that is very specific to individuals in a particular population. So ang most common na nakita ko online for the example of microenvironment would be the spacing between crops, okay, between plants in a bed. Okay, so the spacing of those plants differ. So that is microenvironment. Oh, okay. So the interaction between the genotype and the environment is actually readily seen, mas nakikita siya in quantitative traits. Okay, but before we go to that, differentiate muna natin. Ano ba yung quantitative trait and what is a qualitative trait? For qualitative traits, these are the ones that you are most familiar with when you are taking your basic bio. Okay? Because these are the traits that have been observed by Gregor Mendel. So qualitative traits are traits that fits into distinct categories. Okay? It doesn't appear in a spectrum, it's one or the other. This type of traits are controlled by 1 to 2 major genes. And these are the traits involved in Mendel's experiment. So if you're going to look at these traits on the left side of the screen, so in here you can see that it's differentiated into the traits of the seed, flower, and pod. This is in Pisum sativum. So you have here the form, okay, it's gray and round, or white and wrinkled. So there is a or gray, white, okay, round or wrinkled. So they are distinct with one another. It's either one or the other. It is the cotyledons could be yellow or it could be green. There is no shades of yellow, no shades of green. Okay. Then for flower color, it's white and violet. Okay. For the form, full or constricted, then you have the color of the pod as well, yellow and green. Okay, so these are qualitative traits. When we say naman quantitative traits, these are traits that are not distinct. Ito yung inaaral natin sa quantitative genetics. Okay? The traits, okay, that occurs in or that is presented in a spectrum. Okay? So this has continuous gradations from one extreme to the other. Let's take, for example, our height in humans. Diba? There is the shortest, okay, and the tallest human. And all of us, okay, as a species, is within that spectrum. Okay? Within the range from the shortest to the tallest. So it's not we're just short, or like for example, on the shortest part, and then there's only one option, but to be the tallest. Okay, that's not the case for our height. So this type of traits, ang involved dito are more than 2 genes. So there are a lot of genes that are interacting with one another to be able to determine that specific phenotype. And these are highly influenced by the environment. Mataas yung influence na environment when we talk about quantitative traits. Another example, I think this one is the more applicable for you, is the yield, crop yield. So you have an individual in the population with the lowest yield, and then you have an individual in the population with the highest yield. And the others would fall, okay, from the lowest to the highest. Okay. So here is a table differentiating qualitative from quantitative traits. So for the criterion, you have the difference between phenotypic classes. For a qualitative trait, it is discrete or striking. So it's yellow or green. So there is a distinct classification between these different phenotypes. For quantitative naman, it's not discrete. Again, they fall, or each individual fall, okay, between the range of the two extremes. For phenotypic distribution, discontinuous. Okay, so you just take the number, okay, the specific measurement, or the number rather, of individuals having that particular trait. And then for quantitative, again, continuous gradations from one extreme to another. For the number of controlling genes naman, we have 1 to 2 major genes, and then greater than two minor genes or polygenes. So there are a lot of genes that are involved when you're going to, when you talk about different quantitative traits. And environmental influence, little to none for qualitative traits, while for quantitative traits, mataas yung environmental influence. So for our discussion in genotype and environment interaction, we're going to talk about quantitative. Okay. Now we'll go to the phenotypic variant. This is part of quantitative genetics. A certain phenotype or a certain trait for a quantitative trait, again, nasa spectrum siya, di ba? And it's important for us in crop development or in a development of a particular, be it animal or plant, okay, to know what causes the differences amongst individuals of a population. So, ano nga ba yung cause ng pagkakaiba-iba? Bakit may isang individual in a particular population that yields poorly? Okay? When you talk about yield, bakit merong mas daming mamunga? Okay? And that is what you call the phenotypic variance. Okay? And that phenotype that we're going to talk about is yield. Para lang maging tayo, consistent. Okay, let's talk about yield. In dun sa mga next na slides. Okay? So, this phenotypic variance is the variability of a particular phenotype within a population. The differences in the phenotype. Okay? This is a sample variance for a quantitative trait. So, in quantitative genetics, you're going to encounter statistics, okay, in computing for phenotypic variance. So later, tingnan natin. But of course, the main focus pa rin natin is genotype and environment interaction. Papahapyawan lang natin yung ibang parts. So this includes variance due to genotypic differences between individuals, differences in the genotype of the individuals in a population, variances due to environmental differences. So be it the microenvironment okay, or the macro environment, and variance due to genotype and environment interaction. For us to account, bakit nga pa iba-iba yung phenotype na pinapakita, okay, ng mga individuals of the same population. Okay, so, in the left side, you can see here that you have the parental generation. Okay, this is the genotype. So there are 3 gene pairs involved in, I think this one is skin pigmentation. So there are 3 genes that are involved. One is gene A, B, and then C. And then the phenotype would be very light. And then you have very dark for the for this genotype. Okay. And then for the F1 generation, you have heterozygous for all gene pairs. Okay. Plus, this particular interaction is not complete dominance. So this is actually co-dominance. Okay? There is no dominant allele and there is no recessive allele. Kasi sa basic genetics natin kasi, we are more familiar with dominance interaction that is complete dominance. Diba? Na ma-mask yung effect ng recessive allele. But in this case, this is co-dominance. And then the F1 generation would allow to be breed. And then you have here the F2 generation. And you can see from here that in the F2 generation, there is different gradations when it comes to the skin pigment, from the lightest to the darkest. And then you have here the different individuals, or the number of individuals in the F2 generation that has that particular phenotype. Okay, so of course this is just the proportion, okay, of the individuals in a particular population that has that phenotype. Okay. Let's look at the components of phenotypic variance. So the components of phenotypic variance is the variance caused by the genotype, that is the genotypic variance. Then you have the variance due to the environment, okay? And then you have the variance due to the interaction of the genotype and the environment. So add them together, you will have the phenotypic variance, or the differences in the phenotype. So let's zoom in on the genotypic variance. Genotypic variance is due to several types of gene action. Ito yung variance that is due to the genes. Okay. Due to the different genotypes of the individuals in the population. Okay. And nahati siya into 3. So you have variance due to additive effect, variance due to dominance effect, and then variance due to epistatic effect. Yung genes kasi, there are a lot of interactions within a gene and then interaction with different genes. Okay? Kaya, the sum of all those interactions would make up the genotypic variance. So let's start with the variance due to additive effect. There are some genes kasi that when an allele has more copies, there would be an impact into the phenotype. So that is the additive effect. So let me give you an example. Okay, so for example, let's look at a yield ulit. Okay? So for the yield, it is controlled by 2 genes. Let's say gene A and then gene B. Okay. So in one individual, in one individual, it is heterozygote for gene A, and then um, homozygous recessive for gene B. So let's say that it is homozygous for gene B as well. Okay? So, pagpalagay natin na ang yield ay 7 fruits. So let's say, okay, 7 fruits. 7. Sorry, I'm not used to this style anymore. So yeah. So for the, let's say that the yield would be 7. And then there is another individual that is homozygous for gene A. And then you have recessive as well, homozygous recessive for gene B. And then um, the yield would be 8 fruits. Okay. And then that means that the additional dose of that dominant allele, okay, in gene A caused an increase in the yield. Okay, so kaya siya additive effect. So the more dose of a particular allele is in the genotype, nagkakaroon ng impact doon sa phenotype. There is an increase in the yield of the phenotype. Iba-iba pang phenotype. Yan. Okay? There are different examples. I just made it the yield for it to be more understandable. Okay? So let's say nasa ano naman siya, gene B. So you have the dominant allele for A, homozygous dominant, and then you have the dominant allele for B as well. Okay? Tumaas ulit. It could be 9. Right? So, yun po yung additive effect. Okay? Yun namang variance due to dominance effect, there are certain genes that exhibits complete dominance. So, if there is the presence of the dominant allele in a gene pair, it will mask the effect of the recessive allele. So, those are those genes that are involved or that exhibits dominance effect, okay, are included in this variance. Variance due to those genes. Okay? And then you also have variance due to epistatic effects or interaction genetic variance. Kaya siya V sub I. Okay? Interaction genetic variance. Okay? And this is variance due to genes that exhibits epistasis. So there are certain interactions like non-Mendelian interactions wherein one gene pair, okay, when one of the gene pair has a dominant allele, it will mask the effect of another gene pair. Okay? So that is epistasis, be it dominant or recessive epistasis. One gene pair, once it has the dominant allele, or if it is homozygous recessive for a particular allele, will mask the effect of the other gene pair. Okay? So that is for the genotypic variance. So the sum of the variance due to these different genes following these 3 interactions, that will constitute your variance due to the genotype. And ito lang yung heritable. Okay, ito yung namamana from the parents to the offspring, since this one involves purely the genotype. Okay? For the environment naman, or environmental variance, this is the variance within individual or in the individuals within a population that is due to variable factors such as location, soil fertility level, moisture, season, nutrition, and maternal environment. Okay? So these differences in the environment would cause variations in the phenotype. So that is environmental variance. So you can see here on the right the different computation and how you compute for this 3 I think if this one is environmental variance, then you have the genotypic variance, VE, here. Environmental variance, you have the genotypic variance, and then you have the additive variance. And here you have the different variants of different population. In the field kasi, so if you're going to do a research on, if you want to know the genotypic variance, environmental variance, okay, in a field setting, you need a lot of data. Okay, so you need to get the variance in the parental generation as well as the first filial generation, the second filial generation, and the backcross one generation. So let's say, for example, yield yung titingnan mo. So you need to get the yield of all the individuals of a certain generation. P1, P2, or one of the parental generation, F1, okay, F2, and then backcross 1. Okay? For you to be able to compute for this. Right? So, since 1 hour lang naman yung given sa akin, I will not be presenting you the entirety of the data. I'll give you an example on how to compute for variances. Okay. So, variance. How to compute for variance. Okay. So, you will be given a data set, or if you're going to do it on your own, so you will be developing a pure line, the parental generation, and then you will develop the hybrid, okay, the F1, and then the F2, and then the BC1, and you're going to get the mean of your data set. Okay? So if it's yield, you're going to get the sum of all the yield, okay, and then divide it by the number of individuals. And then subtract the mean from each of the data value and list the differences. So you're going to get the differences, okay, of each of the individual phenotype, okay, divide and subtract it from and subtract the mean from it. Then you're going to square each of the differences from the previous step and make a list of the squares, add the squares from the previous step together, and then subtract 1 from the number of data value you started with. And then divide the sum from step 4 by the number from step 5. So it might be a little bit confusing. So let me just show you an Excel file. So I went ahead and all right, here it is. So this one is an example in Mangbin Quadle. So this one is the one of the parental generation, and it's in centimeters. So ito yung phenotype. One individual is 6.4 centimeter when it comes to the Mangbin Quadle. One is 6.8, one is 6.6, 7, 6, 7.2, and then you have 5.5. Okay? So hanggang dito lang po ito. Hanggang dito lang. So to get the mean, you're going to add everything and then divide it by the number of individuals. So you have 6.5 centimeters. And then 6.5 centimeters, you will subtract from each of this individual data, or each of these individual numbers, or yeah. So you're going to subtract 6.5 from 6.4. So 6.4 minus 6.5, so that is 0.1. And then you will do it for the rest of your data. Okay? And then afterwards, you're going to square. So you're going to square negative 0.1, so that is 0.01. Okay? And then you're going to add the squares. So add all of this, you're going to get 2.1, and then you're going to divide 2.1 by n minus 1. So that is 1 Number of individuals minus 1. So that is 1, 2, 3, 4, 5, 6, 7. Minus 1 is 6. So that is 2.1 minus 6. So that is 0.35. So that's how you're going to get the variance for one generation. Oh, sige, I'll give you time to process. Do you want me to repeat? Ulitin ko ba? Or okay na? Okay, all right, no worries. So you will be given a data set, okay, or you will be making your own data set based on what you have seen on the field. So in this example, you have the mung bean pod length, okay, in centimeters. So the phenotype is mung bean pod length, right? So each of the individual, okay, or individual plant, you're going to measure the pod length. So one would be 6.4 centimeter, the other would be 6.8, 6.6, 7, 6, 7.2, 5.5. Okay? So hanggang dito lang po. Right? So you're going to get the mean, okay, of this value. So you're going to add them and then divided by the number of individuals. So that is 7. So you're going to get 6.5 centimeters. So that is the mean. And then afterwards, you're going to subtract the mean from each individual value. So in this case, 6.4 minus 6.5, so that is 0.1. Then 0.3, 6.8 minus 6.5, that's 0.3. So on and so forth. So that is the difference, okay, so the second step. Okay, so for the third, you're going to square each of this value. Square nito. Okay, so if you're going to square it, so that is a 0.01 and then 1. Okay, and then you're going to add this value. So that is 2.1. And the last, in order to get the variance, is you're going to divide it by n minus 1. Okay? N is the number of individuals that you have accounted for, okay, in a particular generation. Okay? So you go, in this case, we have 7. Okay? So that is 7 minus 1, and that is 6. Okay? So that is 2.1 divided by 6. Right? So that is how you're going to get the variance of P1. Okay na po? Okay. So that is for the variance. So balik tayo dun sa ating balik tayo dito. Yon. So for you to be able to get the environmental variance, you need to get the variance of P1 plus variance of P2, the variance of F1 over 3. So that's how you're going to get the environmental variance, or the effect of the environment into this different into the phenotypic variance. For genotypic variance naman, that is the variance of F2 minus the environmental variance. And then for the additive variance, that is 2 multiplied by the variance of F2 minus the variance of BC1. So since this is actually not the bulk of the lecture, I will just be giving to you the formula, and then you can do it or you can practice on your own. So yeah. And then for the data naman, you can get different publications din naman. You can practice using different data sets coming from different publications when it comes to their quantitative data. And you can try to solve it on your end. Okay po? So that is for the genotypic variance and the environmental variance. So again, these are the differences in the phenotype brought about by differences in the environment and differences in the genotype. Now we'll go to the genotype-environment interaction. This is actually the main topic. Okay? The genotype-environment interaction is present when two or more genotypes respond differently to changes in the environment. Okay? So when you may have a genotype wherein it is homozygous, okay, for that particular gene, or heterozygous. So how they differ in the way they respond to particular environment. So that is the genotype environment interaction. And they could differ in the direction of response. Okay? So if you're going to take a look at yield, okay, a particular genotype responsible for the yield, or the phenotype that is the yield, okay, it can increase, or it can respond positively to a particular environment, or the other way around. Okay? The environment can cause that plant in order to yield more, okay, or an environment could cause that plant to yield less, even though the genotype did not change. Okay, so that is for the direction of the response. It could be, um, it could behave positively or negatively. The degree of the response naman is it could still increase in yield. Okay, it could still behave positively, but um, in a different in a different mm, strength. Okay? Pwedeng sa itong environment na to, mag-i-increase pa rin naman yung yield, pero konti lang. Dun sa isang environment, yung degree of response is larger. Okay? It increased in a huge margin. So that is how you're going to know that there is a genotype environment interaction. So later, mas makita nyo yun with the examples that I'm going to give. Okay? So there are different types of genotype environment interaction. Here, this one is lifted from the paper of Osei et al. So that is genotype by environment interaction, a prerequisite for tomato variety development. So this is the types that we have presented. So the first type is if there is no genotype environment interaction. So paano pag wala? Okay, so let's look at letter A, the graph letter A. Okay, so let's say that the phenotype is yield again. Okay, so there are two environments that we're talking about, E1 and E2. And there are two genotypes that we're going to measure, G1 and then G2. Okay, so will their phenotype change from one environment to the other? So if you're going to look at this graph, okay, G1, or the individual with this genotype represented by G1, is superior or has a higher yield as compared to G2. So taas yung yield niya, okay, as directed by the higher value of it on the y-axis. Okay, so yon, higher siya. In E1, at the same time, in E2 as well. So in E1, G1 still fares better than G2. And then, kahit magbago yung environment, G1 still outperforms G2 in the same intensity. Okay? Even the differences between the yields of the two does not differ in the even though it changed environment. That means that walang genotype environment interaction when it comes to this particular phenotype. Okay? There is, even though nagbago yung environment, okay, hindi nagbago yung value for the yield in both G1 and G2. Okay? Now we'll go to B and C. So this is what you call a non-crossover interaction. Okay? So in graph B, G1 still outperforms G2. However, you can see that there is higher yield in environment 2 as compared to environment 1 in both genotypes. So sa environment 1, you can see lower yung yield as compared to the yield in environment 2. So there is already the interaction between the genotype and the environment. However, okay, you can see that the differences between the two genotypes is still constant, is still consistent rather. Okay? Pareho pa rin yung differences between the yields of the two genotypes as it changed environment. Pareho silang tumaas at the same degree. Okay? And at the same direction then. Both are impacted positively by that change in the environment. Okay? So let's look at C naman. For C, you can see that there is still an increase in the yield, okay, as these two genotypes are being moved from environment 2 environment 2, environment 1 to environment 2. Okay? However, the differences between the two genotypes when it comes to yield is more evident in environment 2 as compared to environment 1. So if you're going to look at E1, mas maliit yung differences between the yield. Okay? The amount of yield, okay, that is exhibited by these two genotypes. But in E2, okay, in the second environment, mas malaki na yung differences between the yield. Okay? G1 outperforms G2 in a larger margin. Okay? But still, it is non-crossover interaction. Wala pa rin namang negative effect yung change of environment or this particular environment into the yield of these two genotypes. Now we'll go to B, right? So for D naman, E and F, this is an example of crossover interaction. So ano ibig sabihin nun? As you can see here, okay, in E1 or the first environment, okay, G2 outperforms G1. Mas mataas yung yield ni G2 as compared to G1. But in the next or in this environment, E2, it's the other way around. G1 outperforms G2. Okay? So their direction of their response to this particular environment is opposite. Okay? They have a different or an opposite response to that change in the environment. Okay? Now we'll go to E. Sa E din, ganun din yung nangyari. So, in the first environment, okay, G2 outperforms G1. Okay? But in the second environment, okay, G1 outperforms G2. But the degree of their response is also different. So in E1, okay, you can see here that there is a small difference between G1 and G2, but in the second environment, mas malaki na yung differences in the yield that is brought by the difference in genotype. Okay? And then the last one, so you have here the in the first, okay, in E1, okay, G2 outperforms G1, okay? And then in the second environment, G1 outperforms G2. And as you can see here, opposite direction din siya, and there is really a huge margin between the two. Okay? Kasi dito class, the trend would still be up. Their response to this environment is still positive kahit na nagkapalit sila ng rank. Diba? Even if G1 outperforms G2, it's still positive. Okay, G2 still has a higher yield in E2 as compared to E1. But for this one, we can see that change in environment caused G2 to have a lower yield as compared to its previous yield. Okay? And G1 have a higher yield in this environment as compared to the first environment. So kita natin how a particular genotype, how a particular phenotype is expressed in an individual having the same genotype but in a different environment. How a particular phenotype is expressed, okay, in an individual that did not change its genotype, okay, but have different or that is grown in different environment. Okay. Of course, if you're going to breed, if you're going to do plant breeding, animal breeding, you have your goal in mind, diba? There are a lot of things, or there is a specific goal in which you want to achieve to develop a particular plant or an animal. Okay? So you will be able to gauge what would be the effect of the change in the environment in the expressivity of the phenotype that you want to be seen or that you want to be shown in your in the variety that you're developing. Okay? So yeah. So what are some of the implications of genotype-environment interaction in crop improvement? So it is necessary for you to test the genotypes on how well they perform in series of environment. Kaya nagkakaroon talaga ng multi-location trials. Diba? So you try to plant a particular seedling in this particular environment and try to have it planted at another location for you to gauge how they fare in those different locations. So it's multi-location, multi-year, or both. So given that there are a lot of environmental factors, so kailangan din nating ma-distinguish ano ba yung environmental factors that you would like to test, be it the temperature, be it the amount of rain, yon. And of course, development of varieties that are more suited to a particular, to different rather, okay, agro-environments or selection of varieties that perform well across multiple sites. So ito yung, again, it will all boil down to your goal, okay, to your objectives when it comes to your breeding program. Okay? So ano ba yung gusto nyo? Do you want to tailor a particular crop that would be suitable or suited to a particular location? O gusto niyo ba mag-develop ng crop that would be suited in a more general environmental condition? Okay? So, yeah. Of course, may pros and cons pa yan. So, it could be if you're going to develop a crop that is suited in a particular location, so you can actually have a higher yield or have a more superior quality of that particular crop as compared to just those that would be suitable uh, to a more varied location. So yeah. So I think I'm still on 5 naman. So I think that's it. So thank you and ace that exam. Ayun.
Yun. Thank you po, Ma'am Jonna, for your lecture, di ba? Ayun. Yes, maraming salamat po. And grabe, napakarami na naman nating natutunan. Isang oras pa lang, parang,
Actually,
kasabog
I'm
na
very
naman ang
worried.
ating ulo.
I'm very worried actually while I am preparing this lecture kasi I'm not sure how I'm going to bring it all in. Kasi if the main bulk kasi dito, di ba, is genotype-environment interaction. But the main concept kasi is quantitative genetics. And quantitative genetics is still, ang part pa rin nun is the genotype, the environment. And isang part lang nun yung genotype and environment interaction. So,
Yes.
and 1 hour lang siya. So, I just condensed everything. So, yeah.
Yes. Kaya nga po, malawak siya, tapos isang part lang talaga ang genotype and environment interaction. So, kung meron po tayong katanungan, ay pwede na po nating tanungin si Ma'am Jonna para masagot na ang ating mga curious minds, di ba? Sino po bang may katanungan?
Are some parts that you'd like me to repeat? I know that I tend to talk in a fast way. So if you have any confusion when it comes to some of the slides, you can actually ask me to repeat. So yeah.
Ayun. Wala na ba? Sige. Habang
so understand na. Ay, wait lang. May sabing wait lang.
wait lang po. Ayun.
Wait po.
Sige.
Sige.
I-analyze natin ng mabuti. Ano ba? May tanong ba ako? Diba? So yun nga, diba? Kapag yes, Sir Felix, meron po ba kayong gustong sabihin?
Hindi, nag-iisip ako ng questions na lumabas last Leia 2021 regarding genotype environment interaction. Pero parang basic lang ata yung tanong about dun eh.
Opo, basic lang naman.
Pero so far, yung discussion naman ay napakalinaw at maayos naman. Kaya,
Ayan po.
So I think
Yun
there is
nga.
a question. Can you please elaborate the difference between additive effect and epistasis? Epistasis kasi, it involves different genes wherein a particular gene pair masks the effect of another gene pair. So for example, let's take into account, I'll just take it up from the top of my head. I'm not sure, I will not sure with the details, but let's take for example, coat color in an animal. Okay, so there are 2 genes that are responsible for that. One would be responsible for the color of that coat. Okay, so be it black or brown. So let's just say, for example, black or brown. And the other gene would be responsible whether or not that particular pigment that would cause that color to be present would be produced. Okay, kung mapoproduce ba siya or hindi. So if, for example, recessive epistasis, if for example, this gene, okay, itong gene pair na to is homozygous recessive, that means that no pigment will be produced. Kahit ano pa yung genotype of the other gene. So kahit na this particular gene dictates that should be black, pero since hindi nga mapoproduce yung pigment, then it would still be albino. 'Di ba? Kasi walang pigment. So that is albino siya. Okay? So that is epistasis. And those genes that are involved or that interacts in that way would be part of the epistatic or the VI, okay, or interaction genetic variance. So yun yung i-account natin doon. Pag naman additive effect, so these are the genes na a particular the alleles that are present will contribute to the intensity of the phenotype. So for example, homozygous dominant, okay, or homozygous yung ating gene, okay, the phenotype would be superior as compared to one that only has one dose of a particular allele or heterozygote. So yun, those genes would then fall under the variance, okay, or that has an additive effect. So iba-iba talaga sila ng interactions. So for dominance naman, yun nga, those genes that are that has a dominance or complete dominance, wherein if there is the dominant allele, it will mask the effect of the recessive allele. So yun, iba-iba talaga kasi yung interaction. There are a lot of interactions between genes, especially if you're going to talk about quantitative traits. Yeah. Hopefully, nakatulong na.
Okay.
Yeah.
Thank you po, ma'am. Yes, di ba? Ang hirap-hirap talaga ng genotype environment or genetics. Diyan talaga ako nahirapan na subject kasi nung college, INC ako. Di ba? So, okay lang yan. Ayun. So maraming salamat po muli, ma'am. Kung wala na pong katanungan, natin po siyang bigyan ng round of applause si ma'am. Thank you po.
Thank you.
Yun. Dami naman nating natutunan ngayong araw. Epistasis. Mga kinalimutan ko ng mga bagay na dapat tandaan pa rin hanggang ngayon. Yes. Kinalimutan ko na sila kasi ang hihirap nilang concept.
Pero kasi sa field talaga, 'di ba? If you're going to do plant breeding, animal breeding, this concept would come in handy for you.
Yes, totoo na po. Kasi based on experience, parang may research yung teacher ko tapos yung professor ko doon sa school yung isang plot tapos doon naman sa isang sa highlands or mas mataas na elevation yung isa, then another doon sa foot of a mountain. So magkakaiba-iba talaga yung areas tapos syempre iba-iba yung effect sa plant. Yun talaga yung naintindihan ko. Diba? At least may natutunan tayo kahit papaano. Okay, again ma'am, thank you very much po Ma'am Jonna Calagua.
Pleasure. If that exam, God bless to everyone that
Yes.
will take it.
Opo.
Sige, I need to go because I have class then after nito. So, thank you for inviting.
Ay, alis ka na ma'am. Ayaw mo na pag-isibigay namin yung certificate.
Sige. Sige.
Silang ma'am, char. Okay, Certificate of Appreciation is proudly presented to Ma'am Jonna Calagua in recognition of her outstanding contribution as volunteer for sharing knowledge during Padayon Hirayo Manaware Free Leia Review Session on February 19, 2022. Signed, Renz J.L. Caducoy, organizer. Signed, Felix S. Valdez, organizer. And signed, Brian Angelo Sustrina, head organizer. Isang parangal clap para kay ma'am. Tan, 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 tan. Woo! Thank you very much, ma'am, and good luck and God bless po.
Thank you as well.
Ayan. So, grabe. Nakakapagod yun na lecture pero dapat marami tayong natutunan dahil genetics ay isa sa pinakamaraming maraming topics ng board exam 2021. Hindi naman natin alam kung anong lalabas sa board exam. So, hopefully, ay may baon-baon na tayo for our 2022 2 board exam. So, ayan. So, ngayon po ay magkakaroon tayo ng online kamustahan. Ito po ay heart-to-heart talk before po tayo po proceed sa ating next na lecturer. Huwag naman po kayong umalis. Gusto po namin kayong makilala para naman malaman namin kung anong mga gusto nyo pang mangyari sa ating online review session. Hindi lamang po kayo mga pumupunta dito at nakikinig, kundi meron po tayong relationship na na-build na kung saan mas makikilala pa po namin kayo at mas makikilala nyo po kami. Kung sino ba kami, kung ano bang ginagawa namin, at bakit namin ginagawa ito para sa inyo. Ako po si Renz, ang inyong kumakandidatong, charot, intergalactic president. So, kinakalaban ko po ang presidente ng Pluto. Charot, baka magalit na kayo. Kuya Bray, okay na po ba ang ating breakout rooms? Malaman ko po kung sino po ang mga taga Luzon. Paki-type naman kung anong area po kayo. Pwede niyo pong i-type sa group chat yung Luzon, Visayas, or Mindanao. So, please paki-checkan naman po sa ating group chat kung saan po kayo galing. Yon! Mabuhay ang Mindanao! Land! Yan. Mabuhay ang Mindanao! Visayas. Sorry, taga Mindanao ako. Kaya, one time.
Mindanao!
Yes! Mindanao. Dito lang kayo sa Mindanao makakakita ng maraming tao na iba't iba ang salita. Sa mga kapatid nating mga Ilocano, naimbag nga aldaw kada kayo amin. Sino po? Ayun, Luzon. Yan. Sa ating mga kapatid na Tagalog, magandang hapon po sa ating lahat. Sa ating mga kapatid na mga Cebuano, saba? Mayong hapon kaninyo. Yes. Hi, Ma'am Cheche. Yun. Hi, Ma'am Avinil Don Eloran. Yes. Mayong hapon. Yes. So,
Ay, Sir
guys,
Renz, pwede
before
na pala
po tayo...
mag-join.
Ay, pwede na ba? Paano po ba?
Pwede po silang pumunta sa breakout rooms.
Ayun, punta
Kung
po
saan
kayo
po
sa
silang
breakout rooms. Opo. Kung saan po ang inyong lugar, Luzon, Visayas, or Mindanao. Huwag po kayong mahiya dahil magkakaintindihan po tayo dito. So,
Tapos sa mga organizers po, mamaya po tayong pumasok sa rooms. Thank you po.
Opo.